Hello everyone and welcome to Deep Inside Biosciences. In this video, we will discuss the preparation strategy for Unit 1 of the Joint CSIR UGC DPT JRF NET examination in Life Sciences and Biotechnology. This unit focuses on the structure and function of biomolecules, which forms the foundation of biochemistry. We will understand what to study, where to focus, the most important topics, recommended books, previous year questions trends, and the best strategy to maximize your score. So without wasting any time, let's get started. Let's begin by understanding the complete syllabus of Unit 1 Structure and Function of Biomolecules. This unit is divided into 10 major topics. First, structure of atoms, molecules, and chemical bonds, covering the structure of atoms, molecules, and chemical bonds. Second, composition, structure, and function of biomolecules, including carbohydrates, lipids, proteins, nucleic acids, and vitamins. The third, stabilizing interactions such as van der Waals forces, electrostatic interaction, hydrogen bonding, and hydrophobic interaction. Fourth, principles of biophysical chemistry, covering pH, buffer, reaction kinetics, thermodynamics, and colligative properties. Fifth, bioenergetic, including glycolysis, oxidative phosphorylation, coupled reaction, group transfers, and biological energy transducers. Sixth, principle of catalysis, including enzymes, enzyme kinetics, enzyme regulation, mechanism of enzyme catalysis, and isozymes. 7. Conformation of proteins, including the Ramachandran plot, secondary structure of motifs, domains, and folds. 8th, conformation of nucleic acids, covering the structural characteristics of aDNA, bDNA, cDNA, and RNA conformations. 9th, stability of biomolecules, focusing on the stability of proteins and nucleic acids. Finally, metabolism, covering carbohydrates, lipids, amino acids, nucleotides, and vitamins. In the next slide, we will discuss the importance and preparation strategy for each of these topics in detail. Now let us understand the complete biochemistry analysis from 2012 to 2025 based on the total of 371 questions asked in CSIR Life Sciences. This table compares every topic using 5 factors: total questions, percentage share, overall importance based on 5-star, frequency, must read or not, where to focus to score mostly in this subject. Topic A is the structure of atoms and molecules. Only 4 questions were asked, contributing approximately 1% of the total. It has a 2-star importance rating and a very low frequency. Therefore, only the basics are required. Focus mainly on the structure of atoms and the structure of molecules. Do not spend excessive preparation time here, but understanding the fundamentals is necessary because they support concepts such as chemical bonding and molecular interactions. Topic B is the composition, structure, and function of biomolecules. This is the highest weighted topic with the 74 questions and a 20% share. It has a 5-star importance rating and an extremely high frequency, making it a highest priority topic. Focus on DNA, amino acids, protein sequencing, globular and fibrous proteins, carbohydrates, and vitamins. You should understand structures, properties, functions, comparisons, and concept-based applications. This topic must be studied thoroughly because approximately 1 out of every 5 biochemistry questions belong to this area. Topic C covers chemical bonds and stabilizing interactions. A total of 16 questions were asked, forming 4% of the paper. It has a 3-star importance rating and medium frequency. This topic is important and should not be ignored. Focus on chemical bonding, hydrogen bonding, ionic interactions, hydrophobic interactions, van der Waals interaction, and the different forces that stabilize biomolecules. These interactions are especially important for understanding protein folding, nucleic acid structure, and molecular recognition. Topic D is the principle of biophysical chemistry. It contributed 24 questions equal to 7% of the total. It carries a 4-star importance rating and appears with a high frequency. So it is a must-read topic. Focus on pH and pKa, buffers, the Henderson-Hasselbalch equation, isoelectric point, molecular charge, ionic strength, mole calculations, and numerical problem solving. This section contains both conceptual and calculation-based questions. So regularly, numerical practice is essential. Topic E is biochemistry. It contributed 61 questions, representing 17% of the total. It has the 5-star rating and extremely high frequency, so it is one of the highest priority topics. Focus on free energy concepts, ATP generation, redox reactions, glycolysis, the TCA cycle, the electron transport chain, oxidative phosphorylation, proton gradient, and chemiosmosis. Do not simply memorize pathways, understand energy changes, regulations, electron flow, ATP yield, and the connection between different metabolic processes. Topic F is the principle of catalysis, mainly enzymes. A total of 71 questions were asked, contributing 19%. It has a 5-star rating and an extremely high frequency, making it another highest priority topic. Focus on enzyme kinetics, enzyme inhibition, enzyme regulation, ligand receptor interactions, catalytic mechanisms, enzyme-based numerical problem, and the interpretation of kinetic graphs. You must be comfortable with Michaelis-Menten concepts, Km, Vmax, different inhibition patterns, and graphical analysis. Biomolecules, bioenergetics, and enzyme together form the most important part of this unit. Topic G is protein conformation. It contributed 36 questions or 10% of the total. It has a 4-star importance rating and high frequency, making it a must-read topic. Focus on 2-dimensional and 3-dimensional protein structure, proteins of the peptide bond, secondary structures, the Ramachandran plot, domains, folds, and structure-function relationship. Learn how amino acid properties influence protein structure and how structural changes affect biological functions. Topic H is nucleic acid conformation. Only 5 questions were asked, contributing approximately 1%. It has a 2-star importance rating and very low frequency. Basic preparation is sufficient. Focus on RNA structure, different DNA conformation, and structural differences between DNA and RNA. Although the direct weightage is low, this concept may also appear in integrated questions with molecular biology. Stability of biomolecules. It contributed 30 questions or 8% of the total. It has a 4-star importance rating and a high frequency, so it must be read topic. Focus on protein folding and denaturations, nucleic acid denaturation, DNA supercoiling, protein stability and protein degradation. Understand the factors affecting stability, including temperature, pH, mutations, solvents, and non-covalent interactions. Topic J is metabolism. A total of 25 questions were asked, contributing 7%. It has a 4-star rating and high frequency, and it is marked as the highest priority area. Focus particularly on amino acid metabolism, nucleotide metabolism, lipid metabolism, pathway integration, and the regulation of metabolic pathway. Question frequently tested the connection between pathways rather than isolated reactions. So understand how carbohydrates, lipids, and amino acid metabolism are coordinated. The final section, topic K, includes hybrid and integrative questions. It contributed 21 questions or 6% of the total. It has a 4-star importance rating and high frequency. This area must be read carefully and requires additional focus because the questions combine biochemistry with molecular biology, cell biology, enzymology, metabolism, genetics, microbiology, immunology, developmental biology, plant physiology, animal physiology, ecology, evolution, and biotechnology. The questions evaluate whether you can connect concepts from different units rather than recall a single fact. The pie chart summarizes this complete distribution. Atoms and molecules 1%, biomolecules 20%, bonds and interaction 4%, biophysical chemistry 7%, bioenergetics 17%, catalysis or enzymes 19%, proteins conformation 10%, nucleic acid conformation 1%, biomolecule stability 8%, metabolism 7%, hybrid or integrative questions asked has 6%. But remember, even though the metabolism part is only 7%, because in bioenergetics, the topics like glycolysis, TCA cycle, ETC, oxidative phosphorylation is there, which is mostly the carbohydrate metabolism part. Due to that, bioenergetics got 17% and metabolism got 7% only. This doesn't mean that metabolism has low rate. You have to focus the metabolism area as well, because it is in and around is equal to bioenergetics. Therefore, the ideal preparation order should be first biomolecules, enzymes, and bioenergetics. Second, protein conformation and biomolecular stability. Third, biophysical chemistry and metabolism. Fourth, hybrid questions and stabilizing interactions. And finally, atoms, molecules, and nucleic acid conformation. This priority-based strategy will allow you to complete the most frequently tested and most scoring areas first. Now let's discuss the best book to prepare Unit 1 Structure and Function of Biomolecules. One of the most trusted and recommended books for CSIR, UGC, DBT, JRF NET is Lellinger Principles of Biochemistry by David L. Nelson and Michael M. Cox. This book provides excellent conceptual clarity, detailed explanation, and high quality illustrations, making it ideal for building a strong foundation in biochemistry. For this unit, you do not need to read the entire book, instead focus on the 16 chapters in sequence. Actually, the chapters here mentioned are not the exact ones, but it is the sequence of topics you must focus on. The first one is water and pH. In that, you have to understand water properties, acid bases, buffers, pH, pKa, and buffer calculations. The second one is amino acids and proteins. In that, you have to learn amino acid classification, proteins, peptide bond formation, and protein functions. The third topic is protein structure. In that, you have to study primary, secondary, structure, and quaternary structures, protein folding, and Ramachandran plot. Coming to the fourth part, enzymes. Covers enzyme classification, enzyme kinetics, Michaelis-Menten equation, inhibition, allosteric regulation, and catalytic mechanisms. The fifth one is carbohydrates. Learn monosaccharides, disaccharides, polysaccharides, glycosidic bonds, and biological importance. Next one is lipids. In that, you have to understand the fatty acids, triglycerides, phospholipids, sphingolipids, steroids, and membrane lipids. The seventh one is membranes. In that, you have to study membrane structure, membrane proteins, transport mechanisms, diffusion, active transport, and membrane fluidity. Eighth one, you have to focus on the nucleotides and nucleic acids. In that, you have to cover nucleotide structure, DNA, RNA, nucleic acid organization, and their biological function. Ninth one, DNA technologies is optional. Read only the basics concept if time permits. 10th one, metabolism overview. Understand the overall principles of metabolism, catabolism, anabolism, and energy flow. In glycolysis, you have to learn every reaction, regulation, ATP generation, and key enzymes. Coming to TCA cycle, understand each intermediate's energy production, regulation, and amphibolic nature. In oxidative phosphorylation, focus on the electron transport chain, proton gradient, chemiosmosis, and ATP synthesis. In photosynthesis, study light reactions, dark reactions, photophosphorylation, and the Calvin cycle. In lipid metabolism, learn beta oxidation, fatty acid synthesis, ketone bodies, and cholesterol metabolism. And the last one, amino acid metabolism. In that, you have to understand transamination, deamination, the urea cycle, and the amino acid biosynthesis. If you complete these chapters thoroughly, you will cover almost entire biochemistry syllabus required for the Unit 1 of the CSIR UGC DBT JRF NET examination. Thank you so much for watching this video till the end. I hope this preparation strategy helps you plan your biochemistry preparation more effectively for the joint CSIR UGC DBT JRF NET examination. The same video is also available in Hindi and Telugu, so you can learn in the language you are most comfortable with. If you have any doubts or if you would like us to make a detailed video on any specific topic, please let us know in the comment section. We regularly create videos based on your suggestions. If you found this video useful, don't forget to like, share, and subscribe to Deep Inside Biosciences. Thank you once again, wishing you all the very best for your preparation and your future research career. Stay focused, stay consistent, and remember, success is in your DNA.